Okay, this one's good. Okay. Good afternoon, everyone. On behalf of agricultureinformation.com, I'd like to invite the speaker, Dr. Sesha Kiran Kollipara, who will talk about biofertilizers and biocontrol agents for sustainable food production. I now request the speaker to begin. Thank you.
Good afternoon, everyone. Thanks for the invitation, madam. I would like to introduce myself. I am Dr. Sesha Kiran Kollipara. I am the Senior Scientist Plant Pathology, and I am currently posted in the Biofertilizers and Biocontrol Production Unit of Dr. YSR Horticultural University at the University Headquarters in Andhra Pradesh. I would like to take you to the audience to each and every step. What are the starting from the introduction and what are our products and the what are the necessity of usage of the biofertilizers and others one by one. One minute. Is my screen visible, madam?
Yes, it is visible.
Yeah. So as I told, biofertilizers and biocontrol agents. So we'll start with my introduction and what are the different biofertilizers available for which are beneficial for the croplands and how they are being used in the cropping systems, their mode of action, and what is their necessity, and what are the different constraints in utilization of these biofertilizers, etc. As you all know, we start with a little bit of introduction. So, why we need for biofertilizers and what are the current situations? So, the urge for enhancing the yield due to increased pressure on the food production has led to the indiscriminate use of chemical fertilizer and other agriculture. As we are aware that during 1960s and 1970s, the food grain production in India is very low, and we are forced to increase the fertilizers through the adoption of the short duration variety, and along that is coupled with the abundant usage of the chemical fertilizers and other agrochemicals. Because of these varieties which are highly responsive to the chemical fertilizer, that has become part and parcel of our farming systems. But there are some other ill effects too. Because of these chemical fertilizer, the soils are getting degraded, and as a result, many of the beneficial and other microorganisms which are present in the soil and which are contributing to the development of the cropland, they are getting deteriorated, and there are many other side effects. For these soils, they are becoming and they are highly dependent on these chemical fertilizers. So in this scenario, we have realized the importance of the soil, and how to and how to enhance the soil fertility, and to so that even the future generations has to be have to use this very precious soil with beneficial microbes for the sustained crop production. So as a biofertilizer, so in this context, not only chemical fertilizer, we have to use the biofertilizers. So how these biofertilizers are different from the normally used chemical fertilizers? As we know, like many of the farmers, we use majority of the farmers, they use NPK, nitrogen based phosphorus and potassium based fertilizers like urea, or single super phosphate, muriate of potash. These are the nutrients, these are the available immediately. So what are these biofertilizers means? Instead, there are some microorganisms which are doing the same function as the chemical fertilizer. So, but these are not chemical; these are bios, means living microorganisms which are which are alternate to the normal chemical fertilizers. So these they act as a suitable alternative to counteract the adverse environmental impacts. As before I said, we are dumping lot of chemical fertilizers to and into the soil. Because of it, the fertility of the soil, for the short-term benefits, we are chemicals. As a result, the soil is polluted, and the crop is So depend only on these chemical fertilizers. So in this context, we are the important biofertilizers arises. So the as I told, different microorganisms, and just like a chemical fertilizer, these also biofertilizers, they promote the growth and yield of the crops, but in an eco-friendly manner. They don't spoil, they don't add any chemical to the soil that will adversely affect the soil. And and these are, as I told, these are different types of fungi or microorganisms, or microorganisms, fungi, bacteria, and others are actinomycetes like that. So they may contain living or dormant microbes, and these biofertilizers either applied to the crop plants, or they apply seeds, or they apply to soil, so that they will apply in the natural environment or rhizosphere of the rhizosphere or phyllosphere of the plants, and so they contribute to the plant growth. So in this context, there is another term called PGPR. This is very regularly being used. Nothing but a plant growth promoting rhizobacteria. Or, excuse me. So as in this context, we say another word. Rhizo means rhizosphere. Rhizosphere is the part of the soil where crop plants have their the root system is occupied. That particular part of that is called rhizosphere. So in that right here, where they are present, there are certain beneficial microorganisms or like bacteria. So they are colonizing the plant root system and doing some great benefit to the plants, and thereby they are contributing to the growth and yield of the crop plants, and they are also protecting from the biotic stress, diseases and insect pests. So these promoting rhizobacteria, they exhibit already with they are doing good things to the plant. So they exhibit certain synergistic means they are they are is synergistic means they are helping with already existing microorganism in a positive. So their presence is only complementary. It is not and it is not plant. So in the same way, and antagonistic interactions. Antagonistic means suppose the soil is have in the rhizosphere, there are certain microorganisms. So these PGPR, they have an antagonistic effect against those organisms which are causing diseases. So in that way, the disease population, the disease causing plant pathogen population is getting reduced by it is indirectly it is helping the plant to grow to prosper well and giving a good yield. So besides, they are not only controlling the disease, not only supplementing nutrients, they are also they are providing the plant growth by facilitating biotic and abiotic tolerance. So in this context, there is another two words there. The plant is subject when the plants are in the environment, subjected to two types of stresses. One is biotic and another is abiotic stress. Biotic means these are the living organisms. Living means they are subjected to certain diseases which are caused by plant pathogen, fungi, bacteria, viruses, and similar there are some other insect pests which are also living. So these are the biotic, and similarly there are abiotic stresses. Abiotic means which are living and they are contributing to they are having a harmful effect on sustenance or performance of the plant. Like they don't they need not be any living organisms. There may be like extreme temperatures or high temperatures or low temperatures or like a drought conditions or like problematic soils, high salts, like high pH, low pH, acidic soils, alkaline soils, like this. These are they don't abiotic stresses, and the presence of these microbes, they help in overcoming the plant to face these biotic and abiotic stresses. In spite of presence of these biotic and abiotic stresses, the plant continues to grow well, and they draw the nutrients. They facilitate the plant with the required nutrients, and that they thereby contributing to the development of the plant. So because of this synergistic growth promoting activities, so and besides these are not chemicals as PGPR, these are just a bacteria, and they provide these are very friendly and a green manner. So and in contrast to hazardous chemical fertilizers which are adding to with those contributing to the development of the plant. They are resulting in the presence of lot of chemical, and they're spoiling the soil. So this is in a biological approach where we are using certain microorganisms. So in the same way, there is a biological fertilizer. Another is biological control. So as I said, like not only these microorganisms, they are contributing to the plant growth. They also help the plant in overcoming certain fungi and bacteria or viruses which help which cause diseases to the plants, and thereby reducing their performance or reducing the yield. So these biocontrol agents, there are certain microorganisms for like Trichoderma, Pseudomonas, Bacillus subtilis, and like that many other microorganisms which act detrimental to the pathogen disease-causing microorganisms. Their action is by both direct and sometimes indirect. So which will go in a detailed way in the later, which we under separate headings like parasitism, antibiosis, or competitions. So, as the previous later, what is the necessity of using biofertilizers? Where in contrast to the already existing chemical-based fertilizers, means this overusage of these synthetic agrochemicals for enhancing the crop productivity has deteriorated the biological chemical health of the arable soil. So they are when they are going in the forest area or like another richer now there are these rhizosphere, they are abundant in plant growth promoting organisms, but in the presence in the due to the intensive or extensive cultivation and dumping of several huge agrochemicals, that is resulting in the death of these beneficial microbes, and also they are altering the physical properties and also the chemical properties like um, the or like the pH. In this context, we need to say generally most of the cropland they will be present, they will be performing well during the pH of 6.5 to 7.5, or like near acidic, or just near alkaline or a neutral pH. But because of these continuous addition of these chemical fertilizers, the pH of the soils alters drastically. Because of the change in the pH, the plant is not able to take up the nutrients, though they are present in the soil. So that is resulting in the decline in the agriculture productivity. This is a alarming situation. Every farming area, whether it is a country or across the globe. So there is and besides, why we have to depend on the biofertilizers, and whether where we should look into the eco-friendly approaches, means currently, as due to the urbanization and due to the increase in the population, and more number of the amount of the land available for agriculture is reducing, and that is the reason we need to go for a healthy approach, and moreover abundant we have to increase the sustained food production for the more number people that have to the per unit area of the land. We have to produce more amount of the so we have to strengthen the soils, or we have to strengthen the soils which have been problematic with microorganisms and increase yields. So in order to fill these escalating demand for the escalating, the yield and productivity of the agriculture needs to be increased along with the production of agriculture related commodity. So previously also I have told, just for repetition, I'm just saying what is the biofertilizers or plants means, they have to be like lot of progress has been in the area of these biofertilizers. So these biofertilizers, which are nothing but plant promoting growth, microorganisms, fungi, or bacteria. So they establish association with the plants, and they contribute in a complementary to the and they complement the growth and productivity. So biofertilizers, these are, as I told, these are different fungi, microorganisms like fungi and bacteria, and these formulations which we supply to the farmers, and these liquid form talc formations, they will be having certain living organisms, which may be in actively growing in a dormant phase, and applied to the soil, or in there they can uh, applied at the base of the plant or in the rhizosphere, or it can be applied on the surface of plants, like plant surfaces. So once they colonize the plant surface phyllosphere, or they may enter into the plant system through the stomata, and they colonize, and they become endophytes, and they to the plant. and they increase the nutrient availability, or they stimulate the plant for the production of certain phytohormones which are doing good to the plant. So these are the general these are nothing but the plant living microorganisms. So as I told, these biofertilizers are supplied previously in the form of talc formulations or like solid formulation. With the availability of the latest technologies and are because of the requirement of the more number of microorganisms per unit, like per gram or per ml. So there are alternatives like liquid based fertilizers, where per ml of the liquid biofertilizers or biocontrol agent has more of the more of the beneficial microorganisms in contrast to the solid formulation. So we are going for advancing for the based biofertilizers. So these are nothing but, as previously I told, biofertilizers means fertilizers. Generally, most of the fertilizers we use for like nitrogen based, phosphorus based, and potassium based. So these biofertilizers are nothing but, so we are using certain microorganisms which help the fix the nitrogen, which the nitrogen is already present in the atmosphere, but the plant is not able to utilize. So these fix the nitrogen by converting that atmospheric nitrogen into ammonical forms or nitrate forms, and so that the plant will be able to utilize those nitrate sources or ammonical sources. In the same way, there are certain phosphate based phosphorus solubilizing fertilizers, the potassium mobilizing fertilizers, which we say PSB and KMB, phosphorus solubilizing. See, these phosphate sources are like potassium sources. They are already present in the soil, but they are in a form which the plant is not able to utilize it. So we supplement the soil with certain bacteria like phosphorus solubilizing bacteria. They solubilize the insoluble forms, and thereby making the plant available to the phosphorus sources. In the same way, the potassium is also present, but it is an unavailable form. So these potash mobilizing bacteria, they dissolve or they make it available to the plant. So in and make it available. So these are the nitrogen fixation bacteria, phosphorus solubilizing bacteria, and potassium mobilizing bacteria. So these are the major three NPK. These are the major nutrients. And if we supplement these three different types of bacteria, the plant requirement of the nitrogen, plant of the phosphorus and potassium is met. So these micro present in the employ several mechanisms to provide crop benefits to the crop plants. They can be either efficient in nitrogen fixation, phosphate solubilization, and plant growth promotion, or possess a combination of all these traits. Besides, these biofertilizers, in addition to this fixing nitrogen or mobilizing the phosphorus or potash, they stimulate the plant to produce certain phytohormones. Like hormones means which are helping in the plant in a positive way, like auxins, gibberellins, and cytokinins, which are involved in the growth of the plant or cell division, or like many other factors, and they increase the nutrient availability. So they can biological fertilizer, they can fix nitrogen through biological nitrogen fixation plants, solubilize nitrogen required plants such as phosphate, zinc, and potassium. As previously I told, only phosphorus and potassium, but there are plants like rice, it needs more of a zinc, and it more when there is zinc deficiency, the plant the productivity is reduced. So there are certain bacteria like zinc solubilizing bacteria. In the same way, like other iron solubilizing bacteria, many important nutrients, those are which are present in the soils in insoluble form or unavailable form, they will be converted into the available forms by in the presence of these different microbiological process of these bacteria, and make the plant available these nutrients which play a critical role in the in the plant development. Besides, they are stimulating the plant to produce plant phytohormones. And previously, as I told, these biofertilizers, they can be either applied to the soil, or they can be treated to the seeds, or they can be applied, sprayed on the plant surface system. So these nutrients can be cycled, and they help in the crop production for the sustainable farming. So what are these advantages over the chemical means? These, unlike the chemicals which are resulting in the soil pollution, water pollution, these microorganisms, they are eco-friendly, and they form sound sources of renewable nutrients required for maintaining soil health and soil biology. So generally, if a chemical fertilizer is applied means only that particular chemical which is available to the plant for a period of 1 week or 2 weeks or 3 weeks, that is available, it is done. But these microorganisms which we are supplying as biofertilizers, they establish themselves in the plant, in the root system or in the surface of the plant, and they continue to do the benefit as long as they are present in the, they get established in the soil or in the surface. Further, as I previously told, these microorganisms, they also exhibit they have a detrimental property, or they exhibit antagonistic activity against several agriculture pathogens, or which are supposed to cause several diseases. And besides, they also help the plant against several abiotic stresses, like extreme temperatures, temperatures, or like drought conditions, or problematic soils, salt even in the presence of more of the salts, the plant continues to grow, or in the drought condition, the plant continues to prosper well without any decline in the yield. So there are different types of microbes which are microorganisms which are commercially available, and they are serving as efficient biofertilizers based on their ability to obtain nutrients from the soil, fix nitrogen, and stimulate the solubilization of the nutrients, and besides acting as a biocontrol agent. So that is and another is the plant growth promoting rhizobacteria. As I previously told, these rhizobacteria, they are present in the soil, and they do a lot of good benefit things to the soil. So this rhizosphere, as just for a repetition, rhizosphere is nothing but the narrow zone of the soil that is surrounding the root system of the growing plants, and which is nothing but a hotspot of the microbial activity in the soil. And these microbes does a lot of benefits by their presence in the rhizosphere area of the soil. So these rhizosphere is colonized normally by a wide variety of microbial organisms, which are like prokaryotes or eukaryotes, like prokaryotes like bacteria, viruses, archaea, and eukaryotes means fungi, means whomist specific fungi, and nematodes, protozoa, algae, and some other insects. So out of these several prokaryotes and eukaryotes, most of them are fungi and bacteria, which are present. They are present in the abundant proportion, and they form a good association with the plant system. So these free living soil bacteria, they are present in the rhizosphere. They colonize aggressively these plant roots, and they promote then they facilitate the plant growth. So, and those bacteria which are associated with with the plant's root system, and they are promoting these are nothing but these are called PGPR, plant growth promoting rhizobacteria. So this term was first used by Klopper and Schroth in 1978. So this type of heterogeneous group of bacteria, and they contribute to the soil microbiome. They produce various types of chemicals in the plant root system vicinity, and they are helping in the plant growth promoting either by nitrogen fixation or nutrient acquisition, or fighting against different types of microorganisms, and also promoting resistance to various abiotic stresses. So these PGPR bacteria, plant growth promoting rhizobacteria, they increase crop yields, they exhibit biocontrol, and besides they prime the resistance system or the immune system of the plants. They secrete some certain proteins, thereby with making the plant immune against or confer resistance against pests and diseases, or they help in the plant to promote nodulation in case of the legumes. They help in the formation of the nodules where the rhizobium will be present, and besides they help in the stimulation of the seedlings to growth promote. So there are different types of plant growth promoting bacteria that have been reported worldwide. Different types of genus like Acinetobacter, Arthrobacter, Bacillus, Frankia, Micrococcus, Rhizobium, Pinibacillus, Klebsiella, Pantoia, Streptomyces, Azospirillum, Azotobacter, Agrobacterium, and Flavobacterium, and Pseudomonas, Bacillus, Thiobacillus, and these are the different types of the extensive list of these different types of plant growth promoting rhizobacteria, out of which generally in any biofertilizer lab, a few are crop specific, are or the requirement specific, like nitrogen specific means azospirillum for a rice, and azotobacter means for a horticulture crops and other crops. For a phosphorus-solubilizing means a certain some certain bacteria, potash mobilizing bacteria, few bacteria, depending on the requirement of the that particular farming systems. So these are multiplied. So, in overview, if you can see, there are different, as we said, there are several plant growth promoting rhizobacteria, plant growth promoting microorganisms. Several microorganisms have been documented worldwide. And if we take one parameter, like as I told previously, abiotic stresses means they help the plants to overcome the abiotic stresses means like drought, extreme temperatures, low temperatures, high temperatures, problematic soil pH like that means the microorganisms contributing to the which are conferring the plant to face the drought and still performance those microorganisms are Pseudomonas fluorescens, Enterobacter and Pinibacillus and like Azospirillum brasiliensis and Rhizobium. So in the same way, another abiotic stress is salinity. So in the presence of more of salts, the plant's performance to continue to yield, to grow and give the required yields reduces. So there are certain bacteria which by their presence help the plant to continue to prosper well in spite of the saline conditions means these they are conferring to the salinity stress. They are conferring tolerance to the salinity stress is Bacillus pumilus, Achromobacter, Azospirillum, Bacillus megatherium. In the same way, like as I told, apart from abiotic stresses like presence of many fungal bacterial diseases, like microorganisms present, they help the plant to uh, to continue to perform in spite of these presence of these pathogenic microbes. These bacteria they secrete certain compounds or antibiotics or like antimicrobial compounds, so thereby killing the pathogens or making the disease causing pathogens inactive. Like these will maybe like Bacillus amyloliquefaciens, Streptomyces, Pinibacillus, Pseudomonas, etc. And whereas the Pantoya and Pinibacillus, they help the plant in increasing the nutrient absorption, which are already present, but these micro present, they help the plant to increase absorb the nutrients. And other microorganisms like Serratia, Pseudomonas fluorescens, they help in the enhancement of the seed germination. And as previously I told, they help in the plant growth by stimulating the plant, or like they are helping the plant. They are by stimulating the plant to produce the required hormones like auxins, gibberellins, and cytokinins. So these bacteria, Azospirillum, Bacillus subtilis, Arthrobacter, and others. So other factors like soil fertility enhancement and the bioremediation, and also they help there are certain bacteria they produce some secondary metabolites. They help the plant to grow properly like Bacillus subtilis and Pseudomonas putida and Bacillus. So different. These are the little bit introduction about the plant growth promoting rhizobacteria. So what are the different ideal characteristics we should have for a bacterium should be labeled as a plant growth rhizobium bacterium means there are certain parameters that need to be fulfilled by those bacterium or that type of bacterial strain so that it can be labeled or like documented as a plant growth promoting rhizobacterial strain. Means so it may be performing a particular bacterium may be performing very well under laboratory condition, but they may fail under the field condition. So in that context, we should see that the bacterium which is microorganisms, plant growth rhizobacter, it should be highly rhizosphere competent and eco-friendly. Once this bacterium is introduced into the soil, it should aggressively colonize the rhizosphere and they should colonize the plant system, root system, and does the required benefit as expected. And moreover, these bacteria there should not be any ecological or it should have not antagonistic to the beneficial other microorganisms which are already present. So it should be eco-friendly. Means it should colonize the plant roots in significant numbers within a and should have a high growth ability and it should colonize very aggressively within a short term. Their population should increase very in an exponential manner. And otherwise, it should be able to promote the plant growth. It should also exhibit a broad spectrum of action. Means that there are certain bacteria that they may be highly specific to certain crop plants. So those cannot be labeled as a plant growth promoting rhizobacteria. So these bacteria should be broad. They should have a broad spectrum of action, and they should colonize several types of crop plants, like all types of crop plants, not any particular crop, like single crop or a few benefit only few crop plants, which would be like irrespective of the crop plant, they should be able to colonize all. And besides, whatever micro plant growth promoting rhizobium which are we are introducing into the soil, it should go in a hand in hand with the already microorganisms, beneficial microorganisms which are already present in the soil. And moreover, as our Indian conditions or many conditions, the crops are subjected to different types of abiotic stresses, or abiotic, like high temperatures or extreme freezing temperatures, like in the case of North Indian condition, like Kashmir conditions or Himachal Pradesh, where the temperatures will be very low under during a particular year or part of the year. In the same way, there are certain like Rajasthan or other area where the temperatures will be very high during the summer. So whatever the microorganisms which we are introducing, that should be able to perform well, irrespective of the climatic conditions, or temperatures, or like drought conditions, or water availability, or the radiation, or the presence of other agrochemicals, or like other soil properties. So it should the introduced microorganisms should be highly better competitive skills over the existing rhizobacterial communities. Those are, as previously I told, the rhizosporium is already laden with a lot of microorganisms. So whatever my plant growth promoter which we are introduced, it should have a good it should compete with the existing strains, and it should perform or outperform the existing strains, and does the benefit to the crop plant. So only when the introduced microorganism fulfill these particular parameters, then only we can call it as an ideal plant growth promoting rhizobium rhizobacteria. So being the dominant rhizosphere microbial community, they are actively or passively involved in the growth promotion. Biofertilizer, they promote the plant growth. They will be facilitating plant growth, biotic and abiotic stress, and supporting the plant nutrition. So the different types of mode of action are there, solubilizing the nutrients, or nitrogen fixation, biocontrol of the diseases, phytohormone productions, like that. And these can be categorized into direct and indirect benefits, which we can see like in a representation in this type of in this picture, where direct effects means they are fixing the nitrogen, solubilizing the nutrients like phosphorus and potash, or zinc solubilizing, or iron solubilizing, like this, and thereby promoting the crop plants. So these are directly doing direct benefit. Other indirect benefits means these rhizobacteria, the these bacteria which when introduced into the soil or when applied onto the plant system, they colonize the phyllosphere or rhizosphere, as the case may be, varying with each plant growth promoting microorganisms. They should be good in they should have a good they should compete very well with the existing rhizosphere microorganisms or phyllosphere microorganisms, and does the required benefit. In the same way, they should exhibit antibiosis. Means they as in the rhizosphere or the phyllosphere. In phyllosphere means the part of the on the leaf surface, there are several types of microorganisms, and that particular area is called phyllosphere. Like how the area which is surrounding the roots, we call it as rhizosphere. In the same way, the area on on the surface of the leaf where the microbes are colonizing, that is called a phyllosphere. So there are several types of microorganisms present on the surface of the leaves. So which were the microorganisms which we are introducing onto the which are applying on the leaves or the plant system or in the root system, they should have certain antibiosis. Means they should be there are certain pathogenic microorganisms like fungi and bacteria. They may present in the soil or on the plant system, and they cause certain diseases. So whatever the bacterium which we are introducing, they should be able to produce certain secondary metabolites or antibiotics, and they should kill the designated harmful pathogenic microorganism, so that the crop is not subjected to diseases, either soil-borne diseases or foliar diseases. So by this antibiosis, so when the crop is overcome by these biotic stresses, automatically the plant will be able to perform well in the absence of these plant pathogenic fungi and bacteria. And besides, as I told previously, where they should prime the immune system, and it should make the plant to produce a resistance related protein, so that the plant by nature, by itself, it will be able to overcome the diseases. So like that, there are previously I told like many phosphorus solubilizing bacteria, zinc solubilizing, like that many are there. And these plant growth promoting rhizobacteria, they help the plant by regulating the phytohormones, like they stimulating the plant to produce auxins, cytokinins and gibberellins, and or ethylene, and they help the plant to grow well. So as indirect effects like suppressing the plant pathogens and other deleterious microorganisms by different mechanisms. These PGPRs, they colonize the pathogenic organisms, and they parasitize the disease-causing microorganisms. They make the nutrients unavailable for the pathogenic microorganisms, and they colonize aggressively in contrast to the disease-causing microorganisms, or they may be producing certain enzymes or antibiotics which have deleterious effects on the disease causing organisms. Like this, multiple multi modes of function, the plant is benefited by against the plant pathogenic microorganisms, and also making the nutrients and the hormones availability. So these are the different types of, and moreover, the ability of the microorganisms to colonize, that also depends on the plant genotype and the stage of the plant, and also the presence of other microbial community, either in the phyllosphere or in the rhizosphere, and the soil type and soil composition, soil pH, all these factors play a major role in the performance of this plant growth promoting rhizobacteria. So this is a small representation of a particular current global fertilizer market, where, as I told, like biofertilizers means majority are nitrogen fixing, phosphorus solubilization, potash mobilization, like that. So in the during 2012, majority of the nitrogen fixing bacteria in the market available are there 77.4%. It is the nitrogen fixing. Phosphorus of solubilizing is 14.6%. There was a little bit change in the after 5 years, like 2017. 79% of the biofertilizers available in the market are like farmers' requirement are nitrogen fixing bacteria. That shows the importance of the nitrogen based fertilizers in the cropping system. And in the same way, like 14% phosphorus solubilizing fertilizers were present in the market. Others means like zinc solubilizing bacteria, like in case of required in the case of like cereals like rice, and other iron solubilizing or like other bacteria, or like rhizobium based fertilizer. So everything is not rosy. There are many challenges and constraints in adoption or cultivate or using the biofertilizers. Like there are regulatory constraints and shelf life constraints, marketing constraints, and in contrast to the other chemical fertilizers, these microorganisms maybe their shelf life is very maybe less. Other chemical fertilizers or chemical pesticides, they have a more shelf life for like many years. But these, since biofertilizers, these are micro based, their lifespan is very short, either 6 months or 1 year. Also depends on the type of carrier we are using. Either like a talc based or liquid based fertilizers, and also the storage of the these biofertilizers, once it is reaching the farmers or during the marketing process. So there is escalating interest in the use of farmers because of awareness, or like because of the department people, agriculture department and horticulture. The farmers are interested in usage of these microbial based biofertilizers. It is constantly increasing. So there, it is also, as I told, there is this usage of the biofertilizer is also associated with several challenges, like from the laboratory to the application in the field level. So they in various cereals and legumes. For developing a new PGPR like a plant growth, it is easy to say theoretically, but to develop a biofertilizer or to identify a particular bacteria or a fungus which is having a crop benefiting properties, that's a very tedious process, and that takes a lot of initial laboratory screening, identify isolating from the soils in the testing in the laboratory, and testing on the crop plants in the under glasshouse conditions and multiple seasons, multiple areas, multiple crop plants. So unless these different types of all the exhaustive procedures are there are met, then only we can develop a effective biofertilizers. So, moreover, those bacteria or microorganisms performing very well under laboratory conditions, unfortunately, may not be able to perform excellently once it reaches the field level conditions, because laboratory conditions and our glasshouse conditions are completely are they are in much contrast to the field level conditions where the farmers are cultivating the croplands. So, moreover, some croplands, some microorganisms, they may not be performing very well under the laboratory conditions. They may be performing very well under the natural field conditions. So, moreover, there are once these microorganisms, biofertilizers, they are having these beneficial properties. They have been identified. These have to be mass multiplied, and they have to be supplied through different sources to the needy farmers. So as I told, different types of constraints are there, like biological regulatory constraints, infrastructural, financial, marketing, and field level quality control and carrier. Carrier based means suppose we have developed the biofertilizers, so we are supplying that biofertilizer like microorganisms in the talc formulation, or like a solid formation, or a liquid or lignite formation, or it can be supplied in a liquid formulation where the microbe count per ml is more in contrast to the solid based formulation. And biosafety. So, and this biosafety means like there may be certain microorganisms which they may be doing good for the plant, but they may have certain antagonistic activities, or like they may be having deleterious effects on the humans. So, those cannot be registered as a biofertilizers in view of the safety of the human beings who are the ultimate end users of the food production, or like croplands. So regulatory constraints and infrastructure. So it is not so easy to, once these biofertilizers they are developed, or some these fungi or bacteria are identified, we need to produce in a large scale, means that needs a lot of infrastructure, funding, and laboratory skilled person, and like that. And to establish a biofertilizer laboratory, it is like from the government organizations like our universities, or like ICR Institute, it is okay. But to implement it by a normal person or industry means that needs a lot of several lakhs or crores of rupees, either for establishing infrastructure, procurement of the fermenters, or laminars, autoclaves, or incubators, or fermenters, and bottling units. This all that invites a lot of funding, and above all, marketing strategies. Today everything depends on the marketing. Mm, uh, so efficient biofertilizer has been developed that it should reach the farmer means it should be properly advertised. It should be made publicly available to through different types of extension agents, or like marketing agencies, through different media like that. So a lot of another quality control carrier like this, many are there. So biological constraints like some microorganisms, they may be performing very well under the laboratory conditions. But once when introduced into the soil, because of the presence of several other microbes in the soil, so in the presence of those other microorganisms, these introduced microorganisms may not be able to compete and perform well and exhibit the required functions like nitrogen fixation or solubilization of the nutrients, or other combating the plant against other diseases and insects, or other abiotic stresses like extreme conditions of temperature, or like salt stress, or like drought conditions like that. So biological constraints and technical constraints, like as I told, compared to the chemical fertilizers, these bioorganisms, these microorganisms, they have a small shelf life, like 6 months or 1 year. Like depending on the carrier we are using, like talc or lignite or liquid formulations, and and during the transit from the production to the end user, they may be subjected to different types of higher temperatures or like storage conditions where they are subject. These are the bacteria, the microorganisms present in the formulations may not perform well, or they may make it dormant, or they get killed during this type of improper handling. So these are the different types of technical constraints and regulatory constraints. So just if we have developed a very good biofertilizers or microorganisms which we have identified by screening and laboratory, it is not just enough. There are certain several regulatory constraints that has to be met before, and we have to register it with government agencies, and we have to provide a lot of data, that it is not it is that said microorganisms as doesn't have any toxicity towards human beings or like ecology, or to the ecosystem, or like it is performing it different multiple crop plants. This particular biofertilizer is having showing its efficacy. It is having broad spectral properties, multiple crop plants, their shelf life, all this has to be met. And all this process, it takes several years to register as a effective biofertilizer. So all these entire legal procedures are for registering a biostrains or screening, development, laboratory, and registering. All this may take several years. So which is complicating the situation, sir. And these constraints, these regulations may be varying from country to country. And as I told, infrastructural complaints, infrastructural constraints. Like to develop a biofertilizer laboratory, we need a lot of resources, like inoculation chambers, multiplication refrigerators, autoclaves, incubators, fermenters, bottling units, and skilled personnel, like all these that we need. And as I told previously, like financial constraints, lack of sufficient financial use is large scale production is a significant drawback, because all this we need a lot of a considerable area for start setting up this laboratory, and skilled personnel and everyone, employment, the workers have to be paid. And these biofertilizers, they are very uh, they are supplied at a very low cost to the farmers, unless there is sufficient subsidy or like promotion through the government. It is not so easy for a private agency to set up and become financially viable. So these are the different types of constraints available. So quality constraints means like there are several regulations wherein we see formulation. There are certain norms, like for every 1 ml, there should be minimum of 1 into 10 to the power of 8 bacteria or fungi per ml or per gram. In case of the solid formulation, it should be having 2 into 10 to the power of 6 colony forming units or bacterial cells per gram. So unless these are the conditions that has to be met for supplying the biofertilizers. So like different types of materials, like charcoal, peat, or like lignite, talc, they are being used for the solid formulations. In the same way, liquid fertilizers are there. As I told, biosafety means there are some biosafety levels are there, where the developed microorganisms should not have any harmful effects, or they should not be any pathogenic to the humans, or any like other pet animals are like that. Like suppose example, Pseudomonas aeruginosa, Pseudomonas putida. These have been identified, isolated from the soils, and they are having excellent biological biofertilizer, biocontrol properties. But they are supposed to cause certain diseases in the human beings, or like lung infections. So in that context, we cannot use those microorganisms for the as a biofertilizers, because they may get into our human system, and they may cause some dangerous diseases, like lung infections or nosocomial infections like that. So this is a short overview of the development of the commercial of the biofertilizers, where in the first we are the plant rhizosphere is laden with several types of microorganisms. Through serial dilution and other procedures, we take the soil, we isolate the fungi or a bacteria, and we do multiplication and then check for a laboratory and greenhouse screening, how they are promoting for the plant system. And then after greenhouse, we'll use it at the field level. And once they are performing well in this one, then we take up a combination of the strains, several types of. So olden days we used only azospirillum, azotobacter, phosphorus. Single bacterium we used to supply as a biofertilizers or a biocontrol. Nowadays, their consortia are being developed wherein two or three different types of beneficial bacteria they are mixed together and supplied to the farmer, so that the farmer need not apply several biofertilizers like nitrogen fixing bacteria, one formulation, phosphorus solubilizing bacteria, another formulation, zinc solubilizing. So instead of this, if multi benefit the different types of microorgan which multiple benefits, they are mixed and they are provided as a consortia, so that the need to purchase multiple biofertilizers is avoided by the end user. So in the same way, product refinement toxicity studies, as I told, biosafety levels are there. These toxicity studies have to be carried out, so that the used by microorganisms has no effect on the human life, or like pet animals, or like other or aquatic life, like that. So but only when all these conditions or regulatory conditions are and sufficient data is generated over a period of time, then only that particular microorganisms is approved or overcome regulatory approval, and then it giving certified. So then only we can supply to the markets. So these are the several guidelines. So these are short overview of the different types of biofertilizers which were manufacturing in our biofertilizer production unit, like azotobacter, phosphobacter inoculant, potassium releasing bacteria, and like a solubilizing bacteria. And these are the in the same way biocontrol agents like trichoderma viridi, talc, phosphine, liquid formulations in the same way. Pseudomonas fluorescens. We conduct training programs, and also we invite several from different government organizations, students, and then NGOs, farmers. They visit our lab, and they see the production of these, and we encourage them to take up these, incorporate these biofertilizer intelligence in their cropping system, so that they get immense benefit at a low cost. So this is the overview of the different biofertilizers are like that. Because of the shortage of the time, I could not focus on the biocontrol agents. Only on biofertilizers I have focused. So in if I get an opportunity, we'll try to cover the biological control agents in a other lecture, like wherein we can cover Trichoderma viridi, Pseudomonas fluorescens, Bacillus subtilis, and other beneficial plant growth, plant disease controlling microorganisms. Madam, am I audible, madam? That concludes my presentation, madam. Hello?
Okay. Thank you, Dr. Sesha Kiran, for the detailed presentation. Now, since we already crossed the time limit, I'll try to take minimum questions.
No, madam.
Yeah, if you can answer a little bit quickly, it will be good. So,
Yes.
yeah. What are the major types of biofertilizers suitable for vegetable and fruit crops?
Yeah, vegetables. Yeah, previously during my presentation, I said nitrogen fixing bacteria are there, particularly for rice like that azospirally. Like for the normal horticulture crops like fruit crops and vegetables, for nitrogen fixation, we are using Astrobacter like that. And for phosphorus solubilizing bacteria, there are certain bacteria which we are using, piece of phosphorus solubilizing bacteria to meet the requirements of the phosphorus. In the same way, potash mobilizing bacteria, those are also being uh, prepared by us. In short, like Astrobacter, phosphorus solubilizing bacteria, and potash mobilizing bacteria. So since
Okay.
majority of the requirement is all the crops is macronutrients and nitrogen, phosphorus, and potash. So this is applicable. These they have a broad spectral usage. The only thing is for rice, nitrogen fixing is azospirillum bacteria. For in case of the other horticulture crops like fruit and vegetable crops, we use Astrobacter species instead of azospirillum.
Okay. And how do biofertilizers compare with chemical fertilizers in terms of yield and soil health?
What, madam? What can you please
How,
repeat?
how do how do biofertilizers compare with chemical fertilizers in terms of yield and soil health?
Yeah, soil health means like chemical fertilizers we add to the soil that cause pollution to the soil like that. But and they give instant result like chemical fertilizers. But these biofertilizers we are supplying in a formulation liquid that like talc or liquid formulation which need to be mass multiplied like talc formations like a trichoderma viridi or like that. They have to be generally trichoderma viridi we apply it to the soil, so they have to mass multiplied in farmyard manure and neem cake over a period of like 1 week or 10 days, and then apply it to the soil. In the same way, liquid fertilizers we add in a liquid fertilizers means like bacteria or like astrobacter or pseudomonas that has to be mass multiplied in cow dung or a dung and jaggery, and so that in because bacteria multiplication is very fast, just in a 3 to 4 days these bacteria can be mass multiplied and then applied to the soil. So compared to the chemical fertilizer, they may not show immediate effect, but this will take a little bit of time to colonize to multiply, to establish themselves in the root system or on the plant system, but they have a long term effect. So chemical fertilizers, once it is applied to the soil within 1 week or 2 weeks, like 3 weeks, the requirement of the nutrient requirement is met. But biofertilizers, they once they are establishing means there is a long they serve they keep continuing the required benefit over a period of time in the established atmosphere, in the rhizosphere or in the phyllosphere.
Okay. And what is the ideal method and timing of biofertilizers application in the field?
Ideal method means yeah, because of time constraint I could not tell, madam. We are applying this in previous question only I have already covered that one. So like talc formations are there. So trichoderma viridi, it is a good excellent biocontrol agent with broad spectral activity, or other liquid solid formations means that has to be multiplied. This trichoderma, it will multiply only in the soil based or in a organic matter presence, so it cannot be applied to the folia. So this means, so and that too, these fungi, they will multiply fast in the solid medium, like so. We advocate talc formations or a liquid formation. We recommend to mix with farmyard manure, say 1 kg of the solid formation. We suggest the farmers to mix with 200 kgs of farmyard manure and 10 kgs of the neem cake, and then suggest the farmers to make it wet adding sprinkling the water on its by adding whatever 200 kgs FIM, 1 kg, 10 kgs neem cake, and 1 kg of the talc or liquid formation lead, and that has to be regularly made wet by sprinkling little bit of water, so that it will mass multiply in a period of 1 week or 10 days. Because it is fungi, the development or mass multiplication will be little slower. But in case of the bacteria, like a phosphorus solubilizing bacteria or a potash mobilizing bacteria, or in case of the biocontrol agents like Pseudomonas, Bacillus, we suggest, because these are bacteria, they multiply very fast. Within a minutes, they will multiply, they will become double. One becomes two, two becomes four, four becomes eight, like that. So because bacterial multiplication is very fast, so and that too, they don't need any solid medium. Just by take a 200 liters vessel, which every farmer will be having. We suggest them to take 180 or 190 liters of water, mix 5 kgs of the dung, 2 kgs of the jaggery, because jaggery becomes carbon source for that will enable the multiplication of the bacteria in a very short period of time. Then we suggest the farmer to add 1 liter of the liquid fertilizer or biocontrol agents to this water, and cover it with a tarpaulin sheet like that, and mix the formulation daily, morning and evening, for 3 or 4 days. So within 3 4 days, this formulation will be very multiplied with a huge number of bacteria, then which can be supplemented to the soil, or by filtering we can be used for foliar spray, so that once these bacteria are applied to the soil or in the foliage, they will establish themselves on the plant system or in the root system, and does the required benefit or does the required job as expected.
Okay. Um, okay. And what are the most effective biocontrol agents for managing common pests and disease in horticulture crops?
Majority, like we deal with diseases, madam. Diseases means soil-borne diseases. Many microorganisms are like Oomycetes fungi, Rhizoctonia, Sclerotium ralphi, like that. So like any which are causing nursery diseases, damping off, or like root rots, dry root rot, wet root, soft rots. Like this, many fungi are there which are causing diseases in horticulture and agriculture crop. For that, we generally recommend Trichoderma viride, which is a majority of the farming communities, they are well aware. This trichoderma establishes very well, um, colonizes very fast, and they, irrespective of the crop, it has a broad spectral activity, like capable of controlling multiple soil-borne diseases, soil-borne pathogens which cause diseases and in several agriculture and horticulture crops. So that is a trichoderma viride, but it has to be applied to the soil. It needs some organic matter for establishing itself, colonizing itself over a period of time, say 1 week or 10 days. So we recommend the farmers in the last plowing, during the last plowing, once the mass multiplied trichoderma viride in FIM and NAME has been supplemented into the soil, so that it establishes by the time the plant the transplanted plants, the transplanted seedlings or the crop plants, they are subjected to different biotic stresses like these pathogenic microorganisms. In the same way, there are Pseudomonas fluorescens, which is a bacterium which can colonize in the root system and also on the plant surface system. So they are available at a very low cost, and and another is the Bacillus subtilis, which not only does the plant growth disease control, but it also stimulates the plants to produce the hormones, has secretes certain good plant growth promoting compounds, like stimulating the plant system for phytohormones. Besides, in contrast to the Trichoderma viride or Pseudomonas, Bacillus subtilis, it will, under extreme conditions, like high temperatures or low temperatures, it has the ability to form endospores and survive even under extreme conditions. So once the normal conditions are returned, these endospores will again turn back to the normal bacteria. So they have the mechanism to form endospores and survive under harsh conditions, like extreme conditions. So that type of beneficial property is not there for Trichoderma viride and Pseudomonas. So this ability to perform under extreme temperatures, ability to survive under higher temperatures, even at 70 or 80 degrees, and surviving in a dormant form, and only upon the return of the congenial conditions, this bacterium will be able to continue to perform. It does plant, it acts as a biostimulant, it acts as a biocontrol agent, it helps in the plant growth promoting, help the plant in overcoming the diseases. So this Bacillus subtilis is there, which has more beneficial than the Trichoderma viride and Pseudomonas. So because of these only, our biofertilizer and biocontrol laboratory, we are synthesizing, we are mass multiplying this Bacillus subtilis, which is being used by the farmers to overcome, particularly in cucurbits, like it helps the plant to protect against powdery mildew and other diseases, which is a major constraints in under winter conditions, where the congenial conditions of low temperatures and high humidity is resulting in this type of diseases. And in that conditions, this Bacillus subtilis performs very well.
Okay. Okay. Yeah. How do biocontrol agents interact with other crop protection sorry, I'll repeat. How do biocontrol agents interact with other crop protection inputs like neem oil or botanical extracts?
So because all these microorganisms, like in case of any other chemicals, means their performance may be hindered, but they go in they are a hand or complementary effect with other non-chemical based, like neem-based formulations, like that. Neem-based formulations or pongamia extracts, or pongamia, or like other plant-based, they don't have any harmful effect, or they don't have any hindrance for the performance of these effect of these biofertilizers.
Okay. And can they be applied through drip irrigation systems?
Yeah, that is the major thing, madam. Generally, most of the formulations in the commercial we say the farmers will, they say the liquid formulation, solid formulation, they say they apply through drip irrigation. But let me make it very clear. These liquid biofertilizers or solid formulation, which we are supplying in a concentrated form. This is a simple way of saying is, this is just like for a curdling of a milk, we add little bit of curd, so, and wait for certain time so that it becomes curd by the next day. In the same way, these biofertilizers are biocontrol agents. These have to be mass multiplied. What we are going with only a starter culture. So this has to be mass multiplied in FIM and neem cake in case of trichoderma and other. In case of the biocontrol, in case of the bacteria means in a liquid container, like we have to add dung and then jaggery, and then wait for 3 to 4 days for development, for mass multiply. Then only we apply to the, recommend to the farmer to apply to the crop. Otherwise, just giving a 1 liter of liquid to the soil and expecting the benefit means it doesn't work. Only because once we are supplying through drip means these biocontrol agents or biofertilizers, they have to be established, means it needs some organic matter for mass multiplying. Suppose we are applying to a crop plants which are already supplemented with a lot of chemical fertilizers, or where there is very less organic matter in the soil. In that conditions, we cannot expect the supplied microorganisms, very few microorganisms, which is going into the root system to do extraordinary job. So the main thing is the supplied biofertilizer should be mass multiplied and then only supplemented to the soil. Then that does a great job, and it does the required benefit, rather than the direct supplying through the liquid in a liquid form through the drip irrigation.
Okay. And are biofertilizers and biocontrol agents easily available to farmers through government or university supported channels?
Yeah, what is the question, madam?
Are
They're
bio
available. Like majority of the ICR institutes, biofertility, they supply the bio in every majority of the state agriculture universities, or in our Andhra Pradesh, like Acharya NG Ranga Agriculture University, our YSR Horticulture University. We have several research stations which are doing this job of supplying the biofertilizers and the biocontrol agents at a very affordable price. Like trichoderma viride, we just supply it to the farmer at mere rupees 120 per kg or 120 rupees per liter. Pseudomonas fluorescens still less, just 80 rupees we supply for the 80 rupees we supply per liter, which is sufficient for 1 acre. In the same way, Bacillus subtilis, this production cost will be a bit high. It needs more of inputs, very costly inputs. So to meet that cost, we supply it at the rate of 300 rupees per liter. In the same way, azospirillum, azotobacter, phosphorus solubilizing bacteria, potash mobilizing bacteria, they are supplied at the mere 100, 300 rupees per liter. And each liter is sufficient to meet the requirements for 1 acre.
Okay. And what is the shelf life of commonly used biofertilizers products and how should they be stored?
Yeah, generally, madam, like we supply liquid formulations and solid formulations. Liquid formulation generally recommended is like it should have they have a 1 year. We add certain diluents like that is having like trihalose, arabinose, and other polyvinyl pyrrolidone, glycerol, like this some other compounds to make the bacterium to be in a live conditions and also supplementing certain nutrient source. So in that conditions, it will be able to survive for 1 year, provided it is properly stored. In the same way, talc formulations, it will be having like 6 months like that. But though it is we take utmost care, and even after manufacturing, we keep in the cold rooms like 20 degrees and store them so that its efficacy, the microbial organisms are performing very well and there is no other harm done. But once it is reaching the farmers, they have sufficient care has to be taken so that it can be it doesn't affect the lifespan of the microorganism. We should not subject under it should be put under some a little bit lower temperatures, not particularly like it should not be refrigerated. Little bit of not extreme conditions of temperature or like that. We should store him in some little bit of shade conditions. Better thing is the new expressions and do it immediately mass multiply and use it directly into the crop plants. Because based on the type of formulation, solid or liquid, the lifespan may vary from 6 months to 1 year.
Right. Okay. And can small and marginal farmers produce their own biofertilizers at the village level?
Yeah, actually, these are very tricky, madam. Tricky mean microorganisms. It needs a lot of skill. Like it should be like because most of the bacteria they look similar, whether we are the required bacterium or other contaminant, other bacteria which are present in the atmosphere. So it is difficult to differentiate by a normal layman. So they can the best thing is, and there no need for development by the farmers. We ourselves we are a government organization. We are supplying at a very low cost. As a tall formations are a liquid formation. And that too, if you want to mass multiply means, as I told during my lecture, it needs a lot of infrastructure, skilled personnel, and who has some ability who has some skill, knowledge to know what particular organism he is handling. So that needs a little bit of knowledge. So better not to take up this manufacturing process by the farmers. These are biofertilizer. These are highly subsidized, and though it is lot of infrastructure is needed, lot of labor is required. Still, the production costs are little bit we supply at a very minimal cost. As previously told, hardly 300 rupees, like azospirillum or azotobacter or phosphorus solubilizing bacteria. Even trichoderma, hardly 120 rupees per kg. Pseudomonas, 80 rupees. So there is no need for farmers to try their hand in multiplying. Instead, if they can use the biofertilizers or biocontrol agents, which we are supplying at a very affordable cost, they can mass multiply and use it in the this one.
Okay. So now the last question. What support or training does Dr. YSR Horticulture University offer to farmers interested in using bio inputs?
Yeah, many of the in the last slides I shown some of the pictures, madam. Regularly, some college students and even farmers, NGOs, they visit our lab, and we provide them all these. We explain to them, and we give the training programs through the Krishi Vignan Kendras, and under Krishi Vignan Kendra, like KVKs, they are ICR sponsored, so there are several on-farm trials and farm level demonstration. Then there are several programs through which these biofertilizers, biocontrol, they are made available or supplied free to the farmers under different programs. So like we give training, we through the departmental, like agriculture department and horticulture department, before the cropping season starts, like all the farmers and all the scientists, they tour different villages every day per week, at least 2 3 days, 2 days, along with the scientists, along with agriculture department officials and horticulture department horticulture officers. We conduct different visits and conduct at least 3 to 4 hours meetings at field levels itself, and make them create awareness, and also make the farmers explain about different benefits of these biofertilizers and biocontrol, and suggest them to make these part of the cropping system. We supply at a very low cost. And to take a given example, in the last, like our cropping season started in the June season, say last June, last month. Hardly today is the 25th. In these 55 days, we have so much familiarized and brought to the notice of the coconut farmers, rice farmers, horticulture, oil palm farmers, like that. In this 55 days itself, we have provided several liters or hundreds of liters and hundreds of cages, and we it was almost, I think, 96,000 rupees worth products. We have made our biofertilizers and biocontrol agents reach the farms. That itself is a I feel personally that we have reached maximum number of farmers, multiple horticulture, multiple areas, even far off places. They suggest, they approach us and they say, sir, we are having a small farm, our income levels are low, and we want to use biofertilizers, biocontrol agents. We supply to them and make them part of the this one. All non-pesticidal management and non-chemical based farming we promote. And even our government itself is very much interested in promoting this natural farming. Andhra Pradesh, it has formed conserve natural farming to protect the ecology, or to reduce the usage of the pesticides or chemical based fertilizers. It is promoting organic farming. Even previously, it was having some zero budget natural farming big project involving several thousands of crores. There are several farmers, hundred thousands of farmers have been benefited, and our biofertilizers and biocontrol agents reaching the people. Even our final year students in the graduation program, in the rural horticulture work experience program, they do a lot of demonstrations, and then they make popularize our products
Okay.
through Krishi Vignan Kendras, through training programs. We supply either free or like we provide at a very highly subsidized rates.
Okay.
Still, if the farmers need more information, they can reach me. Like my number is 97175 or they can send an email. Sesha Kiran, SESHA, Sesha Kiran, KIRAN dot Kollipara, KOLLIPARA @gmail.com. So my number, previously given number, that is having link to WhatsApp. So more information you can reach me. If it is in our nearby vicinity, means we will try to send the required biofertilizers at the given price.
Okay. Okay, Dr. Sesha Kiran. So now we have come to end of question round. On behalf of agricultureinformation.com, we like to thank you for a very detailed presentation and answering all the questions in depth. And we also like to thank our editorial team for sharing the questions. So we'll be ending this meeting now. Have a good weekend and great week ahead.
Thank you so much, madam. I
Thank
think we have given good number of lectures, and thank you for providing me the opportunity to reach a number of students, farmers, and entrepreneurs, and familiarizing myself with a lot of people. I am highly
Yeah,
thankful to the
thank you.
agricultureinformation.com for this providing, and I keep in touch with all these agriculture information and do more service to the beneficiaries. Thank you once
Yeah.
again.
Okay. Thank you. Thank you.